The overall goal of this procedure is to describe how to accurately analyze endotoxin and DNA content of extracted airborne particles sampled on filters.
This method can help answer key questions in indoor and outdoor biological aerosol research, such as understanding health risks. Demonstrating the filter sampling procedure will be Yinon Mazar, a graduate student from my laboratory.
For high-volume sampling, use filters made of quartz fiber, or choose the specific type of filter, as well as a filter cutoff size that best fits the research needs. Pre-bake the filters intended for organic and biological compound sampling by first wrapping them individually in aluminum foil, and then baking them in a laboratory furnace at 450 degrees Celsius for at least 5 hours. This will destroy any pre-existing organic residues. Store the baked filters at -20 degrees Celsius until they are to be used for sampling. Next, clean and disinfect the high-volume sampler, such as the one described in the accompanying text protocol. Then, place the pre-baked filter into the filter cassette and run the sampler in accordance with manufacturer's instructions to collect an appropriate sample. In a Class II biosafety cabinet, cut 22 circular pieces of clean pre-baked filter material using a clean 1.12 cm diameter cork borer. Use these pieces to prepare a range of endotoxin spiked filters to be used as controls.